So let me switch on the camera. Okay.
Yeah.
Yes.
Okay. Shall I begin?
No. It's a small introduction I would like to give. Yeah.
Okay.
Yeah. So, dear participants, hope you enjoyed the inaugural session conducted by AICT as well. I see now 69 people joining back. In the morning, I could see 90 people joining. So we'd like to conduct all the sessions in time, but of course, there may be certain technical snags sometimes, which may be beyond our control. So because the lectures are quite long, around 2 hours each, and all the speakers are actually very busy, I have to really thank them for finding out some time and address all of you. So let us start with the inaugural lecture of this workshop on genome editing for environment. So the first speaker is Professor Krishnamurty Kannan. You have already heard him in the inaugural session. So he will speak on the topic Introduction to Genome Organization and Genetic Engineering. So we have allowed some flexibility to the speakers to choose it, give a title of the topic as per their own, expertise. And Professor Kannan has chosen to speak on the genome engineering and architecture. Before he starts his speech, let me introduce Professor Krishnamurty Kannan very briefly. He is a very unique person who has swapped the job between government and private companies in the biotech sector when there were no any much career in the private sector. And he has been in Span Diagnostics, he has been in Ranbaxy, and many other such companies, biotech companies or pharmaceutical companies in the past. And in between, he would sneak into the government system, whether it's a CSI lab or a university. And finally, he ended up as the Vice Chancellor of Nagaland University, which is the central government university. During his tenure in Nagaland as Vice Chancellor, he was the first Vice Chancellor to introduce the School of Engineering and Technology in Nagaland, and then also management education. These are some of the accomplishments of Professor Kannan in the field of academics as an administrator. But as a teacher, he has been very keen on teaching biotechnology as well as developing course curricula for various levels. He started his teaching job in the University of Pune, and he was one of the founder of the course for postgraduate degree in biotechnology funded by DBT. Then after many years, when I was his PhD student in Delhi, I could see him work so hard day and night, developing the course curriculum for NCERT in the course or in the subject biotechnology. In fact, he's a pioneer in that field. He introduced biotechnology as a mainstream subject in class 11 and 12. And for that, he wrote the textbooks himself, and he did that almost, I think, just within a year of its start. So there are many things I can speak to you about Professor Kannan, and he was also the founder, dean, and first professor of Department of Biotechnology in the newly established Indraprastha University in 1998, which was later renamed as Guru Gobind Singh Indraprastha University. And in the area of research, his contribution to the role of ubiquitin and its extracellular role in binding stem cells has been widely acclaimed, not only in the scientific community. It is also part of the medical textbooks, and the media has also covered his work extensively. Currently, Professor Kannan has started KK Biotech, through which he is trying to bridge the gap between academics, reality, and employability, which is, I think, very, very unique. As academicians, we always focus on teaching and research and sidelining the employability angle. I think employability is very, very important to empower the students and build future careers for them, as well as strengthen the economy of this country. So this KK Biotech is quite something interesting. You can find out the details of KK Biotech in the website kkbiotech.net, and you can see that website being displayed here in this slide as well. In fact, just recently, I attended one of the online lectures which he organized through KK Biotech on the COVID pandemic. And all the speakers he chooses are world class, mostly from abroad. And I think many of you can take opportunities to listening to those series of lectures by experts in various areas. So without taking much time, I can see now around 79 people have joined. I request Professor Kannan to start his lecture on genome engineering and architecture. Thank you, all of you.
Yeah. Thank you, Professor Bora, for a generous introduction. So today I thought I'll take you through a journey which I understood, because I belong to a generation who were never taught any of these things. Okay, so many things I have learned on my own. So you might find that a bit strange the way I communicate things, but I communicate what I have understood. So I never communicate something which I haven't understood. So please bear with me, and if you have any doubt, feel free to stop so that I can answer your doubt. So let me begin. So as you can see, basically you have to understand. 1 minute. 1 minute. Yeah. So you have to understand that there is a chemical system and a physical system. Okay. And you have to understand that what are the rules governing, because after all the forces we talk about is electrostatic or hydrophobic interaction, or hydrogen bonding, all these things you hear. But these are all interactions. But this interaction leads to patterns. Okay. And we know that physical laws are operated. But then what is so unique about biological systems? Okay. Even they obey the laws of physics, but in addition to these laws, the physiological and behavioral interaction among the living components are influenced by the genetically and epigenetically controlled properties of the components. So please understand that biological systems are much more complex than chemical systems. In particular, the subunits in biological system, what they call today as system biology, essentially arose out of this. The subunits in biological systems acquire information about the local properties of the system. Okay, and I'll demonstrate you some beautiful experiments as we go along, which will reinforce what I'm trying to say and behave according to the particular genetic programs that have been subjected to natural selection. Remember, a cell is only a vehicle carrying the genetic program, whereas the genetic program is the master program which is written. So this adds an extra dimension to self-organization, which I'll keep emphasizing, because a lot of people say, I created an eye, I created a muscle. These are all self-organizing them. You just put the right condition, it happens. It automatically organizes. So when people say, I did 3D printing and I created a kidney, they didn't do anything. You just put the right component and the organ is made. So that's the beauty of biology. That's why it's a self-organizing system. It's an internal property, not controlled by us. While we make a car, we introduce component, but in biological system, it happens, and I'll talk a bit more on this, because this is what is very important when we talk of how is the genome architecture playing in this. This adds an extra dimension to self-organization biological, because in this system, selection can be finely tuned the rules of interaction. And I'll show you some beautiful experiments which have been done in the 19th century, okay, and 20th century. By tuning the rules, selection shapes the pattern that are formed, and thus the products of group activity can be adapted. So interesting to see simple interactions create complex structures like tissues and organs in the human body. Okay. So please understand that there is a what is called self-organizing system, and that is what you must appreciate. Okay. And I'll talk more about it a bit later. I think it has got hung.
Yeah, your slide is not moving. No problem, sir. You can just wait.
Is there any problem? Now it is moved. Yeah. Okay. Yeah. So what is a self-organizing system? Technological systems become organized by commands from outside, like we make a car and all that, as when human intentions lead to the building of structures or machines. But many natural systems become structured by their own internal process. We are not doing anything. Please remember, when you have a fertilized egg and it gives rise to a chicken, it is an internal process. We only provide the right temperature. We may be incubating at the right temperature, or the animal sits on it to provide this thing, but we are not creating that. A lot of people claim, especially today in tissue engineering, I hear, oh, we published this work, we did. They're all natural. So don't get excited by the claims people make, because it's an internal process. And these are the self-organizing system, and emergence of order within them is a complex phenomenon that intrigues scientists from all disciplines. In self-organizing system, pattern formation occurs through interactions internal to the system. And I'll show you some beautiful experiment in a shorter, but keep this in mind because I can do that by just shifting some cells and create something very interesting without intervention by external directing influences. Self-organization is a process in which patterns at the global level of a system emerges solely from the numerous interactions at the lower components of the system. For example, you look at our own body, you have eyes, you have a brain, you have a mouth. They all are different parts, but they are all made up of the basic cell. And how does that get organized? That's a local event. Okay. And you will see that the whole body pattern is happening like that by self-organizing system. Okay. And how it happens, I'll tell in a few minutes. Okay. Moreover, the rules specifying interaction among the system components are executed using local information without reference to the global pattern. In systems, by an external ordering influence, pattern is a particular organized arrangement of objects in space or time. Remember how the organization, like what happens first, it's a time-dependent phenomenon. Okay, and pattern is a organized arrangement of space and time. So self-organized are not reversible. Okay, until 2006, somebody demonstrated they can reverse the whole process. Why is it all the time getting hung? Utpal?
Yes, sir.
Yeah,
it will
suddenly
move. It will take time, don't worry.
Okay, okay. So progress of knowledge is often the result of, sorry. Suddenly it's a very slow process. Okay. So basically what we are trying to say, progress in any field is happening. So there are three principles on which all our modern biology stand. One is conceive the cell as the smallest autonomous unit of life. Okay. So for life to occur, you need a cell. According to this concept, cells not only constitute the body of bacteria, protista, and complex higher organisms, but they serve as also as vehicles of propagation such as spores, sperms, and eggs. The most important conceptual advance made by Rudolf Virchow of the cell theory was the insight that new cells originate from the old ones. So nobody creates life, it always emerges from the old one. Actually, Robert Remak, who deserves most of the credit for discovering the details of the origin and formation of cell. Many of us don't quote, everybody quotes only Virchow. Remak and Virchow were students of Johannes Muller in Berlin. So you will see the German school played a major part. By 1855, Remak had stated repeatedly his conviction that cells derive themselves from other cells by binary division and provide ample evidence to support his claim. It was Virchow who popularized this in his famous work on cellular pathology in 1858, without even mentioning the work of Remak. So these are all things we'll always hear. Again, it is getting stuck. What to do, Utpal?
So shall I share the slide from this end? You can tell. Shall we try that, sir? Or you are sharing the file or the screen, sir?
I'm sharing the screen.
So yeah, so if you share the file, that will be better, sir.
No, I'm sharing the, no, what I'm saying is, how do I go back?
So you go to below share.
Now it has come. It's
Okay,
a
fine.
bit slow. I mean,
Fine.
so one is cell, the second is Mendelian paradigm. The idea is substantiated by the Mendelian paradigm. According to Mendel's idea, heritable traits are based on units, which he called as, today we call it a gene. Okay. The most seminal concept was that these units come in pairs. That was his great work. And various alleles, models, differing elements, coexisting hybrids, etc. Okay. So all of you are everywhere, and thus the principle of segregation and independent assortment were established. All that. Okay. So Mendel's contribution was the foundation of the other one. So cell, Mendelian paradigm, and the third was the Darwin's great work. Okay. Now, how do you put all these things? So central to Darwin's thinking was to reject the essentialist view of unchanging species. Today, you are seeing it with coronavirus, it is changing constantly. But at that time, everybody said, all the people before him said, look, it's an unchanging species. The species were seen as the targets of a continuous experiment. So, the species were being targeted for change, what we call today as mutation, and which we observe as the process of selective adaptation. Okay. The Darwinian concept of evolution was a challenge for the young cell theory because they thought cell is a constant. They didn't realize that the cell is changing. Actually, what is changing is only the genetic material inside the cell, which is relating to the nature of the cell produce. So, August Weismann, after he had rejected all mechanism with the concept of instructive adaptation, postulated the cellular continuity of the germ plasm as a separate entity from where the mortal somatoplasm is derived generation after generation. He later identified the germ plasm as chromosome, that is germline genome of organs. So as you can see, till 19th century, ideas were being crystallized. But one of the greatest people at that time was Theodore Boveri, who came into this scene. He was a medical doctor who got interested in this thing, and he did some beautiful work on ascaris as well as on sea urchin, and I will talk about it. So he continued his guru's work, Hertwig. There were two brothers who had done some beautiful experiments sea urchin, which he used beautifully to understand. So Boveri's contribution to cell death. So he was the first man along similar parallelly. Sutton also did this. So Boveri's contribution to cell division cycle concept. The comprehensive cell studies of Boveri revealed to him cyclical process during cleavage division. So mitosis, meiosis, all these things what we talk today was actually observed in ascaris by Boveri. Okay. So he could talk about, and he did some very beautiful experiment, whether cytoplasm is important or something else is important. These are all the things. So the chromosome cycle was the one he postulated, and he also talked about centrosome cycle. Okay. Moreover, events commonly associated with fertilization also gave a new insight into the control of cell division. So, what is chromosome cycle? Okay, that although it is generally accepted in Bowery's time that cells are generated by binary fission of a mother cell, it was not clear how chromatin, that is what we call, today we talk chromatin loosely, but it was originally proposed by Weissman's germplasm. He had proposed that is located in the cell nucleus is transmitted to the daughter nuclei such that both are genetically identical to the nucleus from which they arise. The main problem concerned Walter Fleming's nuclear metamorphosis, that is, the transformation of a nucleus into a dancing mitotic chromosome. Because if you look at mitosis, various steps, and meiosis and all that, it is literally a dance being performed, and back to mitotic chroma and back into interphase daughter nuclei. The change of chromatin into threads, okay? So mitosis means thread, okay? Inspired the term mitosis, but did not explain how these elements already anticipated as the vehicles are precisely reproduced and distributed to the cell. So his fundamental problem was, he solved this problem by demonstrating that chromosomes are permanent organelles which are condensed in mitosis and dispersed during interphase. This was where he did it somewhere around 1902. Okay, so 1888 to 1904 he did all this work. But besides his observation concerning continuity and individuality of chromosome, important arguments gathered in support of chromosome theory of inheritance. Bowery gave a truly modern description of the establishment of mitotic apparatus and the interplay of its components to accomplish the distribution of daughter chromosome to the opposite poles. So as you can see, when you talk of genome architecture, what stage are you talking about? That becomes very important. That's why I'm just revising you that many times we just teach it mechanically, mitosis this happened, meiosis happened, but you must know in what system they were doing. And the work of both, Bowery did it on, Ascaris, whereas Sutton did it in the grasshopper. From the detailed observation of cleavage cycles in Ascaris and sea urchin embryos, including multipolar mitosis, half spindles, and monopolar mitosis, which he first observed as spontaneously occurring abnormal and later was able to introduce this thing. So Bowery arrived at 3 rules of chromosome distribution due to his work. In mitosis, chromosomes are bipartite and display a sideness such that astral micro, the man had observed whatever today we talk in chromosome architecture, that man had identified astral microtubules, the radii of the astrosphere can connect at the pole side, the concept that a mother chromosome splits into 2, and this will come to know when we will also talk about stem cells. We'll talk in this thing, mother stem cell. So concept that a mother chromosome splits into 2 and only 2 chromatids which are consistently distributed to opposite poles. These are all observed by him and published. A vision of the not yet discovered kinetochores and at opposite sides of chromatid, which he first termed the chromosomal narrow side. With respect to the special attachment sites of spindle fibers, Bowery distinguished 2 types of chromosomes, equipped with either a localized centromere in sea urchin or a diffuse centromere in Ascaris. Nothing is known about these factors which establish the chromosome sideness, that is, handedness of the chromosome along their entire length. So August Robert had attempted to, now this is another very beautiful experiment which was attempted and is very interesting, that they tried to transform the nucleus of a fertilized toad egg into the nucleus of a fertilized egg of a frog. And this is what later somebody else did in 1960s, which we'll come to in a shorter, and for which he got the Nobel Prize in 2012. Bowery settled on sea urchin eggs as the experiment, because the beauty of this Bowery sea urchin was if you shake it, the nucleus comes off. Okay? Because his mentor had shown that the results, the series of experiments he had performed, but he did not continue. That they shook sea urchin eggs, these eggs sometimes produce fragments that lack a nucleus, and they further demonstrated that these fragments could be fertilized and subsequently showed signs of cleavage. Okay. So that's called the famous Maragon experiment. You can go and read about it. Okay. Now, when all these ideas were coming, the scientific community was not willing to accept. Okay. And you will always see that there is a lot of opposition to the ideas and a lot of critical objects. So Darwin was very much aware of this criticism. Okay. Because he said, because he proposed the theory of descent with modification. So he was the one who was under tremendous attack those days because it questioned the whole the evolution itself. Okay. So you are all familiar with this, so I won't go into much of it. Now you can see this is one of the very classical experiment called Mangold experiment. And how this was done was, and what I'm going to illustrate, that this was an experiment which demonstrated for the first time they could create a bi-headed salamander. Okay. And before that, they will only produce one-headed salamander. So how did Mangold manage to do it is something very, very important for you to appreciate. And I'll just show you how this experiment was done by her. So as you can see, a cell a fertilized egg divides, one becomes two. You can separate the two eggs. So for example, you take a mouse and you take the two eggs and separate the two eggs, and he did it, you can separate it very gently. Even with a baby's hair, you can separate the two and put it into two other, mothers so that they develop. You will get identical. So 2-cell stage, 4-cell stage, you can always produce the whole organism. But the moment you come to 8-cell stage, the ability loses. But these experiments were done on salamander. So what they observed was, as you know, when the cell divides, it keeps on producing, and there's a stage called gastrula. Okay. In gastrula, what happens? The cell starts moving around. Just to give you a basic idea that, whenever an egg fertilizes, you take any egg, what happens? The whole egg rotates by 30 degrees. Okay. And it shows what is called a blastopore, as you can see, which is shown there. So what they did was that they took some of the cells from the blastopore. So the experiment was following. You had what is called a colored salamander and you had a white salamander. So if you take the colored salamander, okay, and take the cells near the blastopore and put it on the other side of the white one, okay, colored one, you have taken this thing, blastopore cell, and then put it on the ventral side of the white one, which is at the gastrula stage. What they observed was, after some time, it produced a bi-headed salamander. Now, you might think that one will be colored and one will be white, but actually what one observes is both are white. So, even if we take the colored blastopore and you transplant it on the other side, so in other words, the cells are being instructed what to become. So in gastrulation, the cells move from one point to the other and that decides. So this blastopore seems to be releasing something which decides the fate of the cell. Okay. And this is the basic, and this is called the Spearman-Mangold organizer. Mangold unfortunately died very early after the experiment, but Spearman developed it further and he got a Nobel Prize for this work, and all the development is based on that. So organizer graph results in induction of a secondary axis. Now why I am trying to say is that you are studying a cell, it becomes very important to know the cell per se doesn't have the capacity to become anything. It is only the cells which are in the environment, which are there, like for example, in this case, the blastopore cells, they release something and that results in the fate of the cell. So fate of the cell is defined by the microenvironment in which it is. Okay. So this is very, very important to show that the cell per se doesn't know what to become. It is like all of us as children, we go to school, then in the 9th class we do. So nobody knows who is going to become what. It is the environment, the teacher who influences what we become. So this is precisely what this is trying to show. I hope all of you could follow what I'm saying. Hello? Can people respond, Utpal?
So yes, sir. Yeah, I
Okay.
think yes, sir. Poonam Sen is writing yes, sir, in the
And
chat.
all of you should remember, the similar experiment was done by ATAL Brownie about in 1909, okay, in Hydra. Okay, unfortunately, this is not so much. He did it in Columbia University, and Mangold probably took that idea from ATAL Brownie's work. There's a lot of debate over it. But Spearman didn't quote Ethel Brown because Ethel Brown didn't show, because Spearman had created the eye and she did not quote that induction phenomena. So there are a lot of politics around it. That's not our concern. But I want you to acknowledge that Ethel Brownie did it in Hydra, whereas Mangold and Spearman did it in salamander. Okay, and they could biheaded salamander. And this blastopore region is called the Spearman's organ. That is, they send signals depending upon its concentration and the time. It will decide you will become the eye, you will become the brain, you will become the nose, you will become the heart. All that programming is done at that stage. That is why it is not the birth which is important, it is not the death which is important, but gastrulation is the most important. If anything goes there wrong, then you run into a problem. Okay. So for Bovary, the possibility of fertilizing, and so now another experiment which he had to show whether, that is one set experiment, but I also want you to recall the sea urchin experiment. So by shaking, he could remove the nucleus, and he wanted to show whether the nucleus is important or cytoplasm. That is to address the problem of nuclear versus cytoplasmic control of cellular and embryonic characters. Okay, the sperm provides a natural delivery system for the nucleus. Okay, so to cut a long story, this thing. So what he did, he chose two species of sea urchin. One is called the echinus, okay, and another spiriechinus. They look morphologically different. So if you take the echinus, remove the nucleus out of it, and take the nucleus out of the spiriechinus and put it onto the echinus one, which has been this thing, he could create he could convert the echinus into the spiriechinus, showing that the nuclear material is responsible. Of course, the success rate was very less, 1 in 1,000 only it happened. But he demonstrated that the genetic information is contained. So many of us don't teach all these things. So I'm just trying to, as a teacher, you should be aware that these are all experiments done in the early years. Okay, later scientists improved this one because now you have micro injection devices and all that, and the success rate's very high. You remember I talked about the toad kind of experiment. So John Gurdon did in a species called Xenopus, and he also transferred, he took the epithelial cells, removed the nucleus, and then he took the nucleus out of it and put it onto a fertilized egg, and you could create the whole Xenopus. Okay, and this work here, so this is what became what is known as the very classical experiment of creating a whole organism. Okay, so that part becomes very, very important, and he got the Nobel Prize in 2012. And the famous Ian Wilmot's experiment of creation of Dolly. All these are similar. So it started with Bovary. It went on, a lot of people tried, then John Gurdon perfected the experiment, then Ian Wilmot experimented. So you can create a whole new organisms from the nucleus, showing the nuclear material is most important. Okay. Now I'm going to introduce you another concept, which is very unique to multicellular organisms. Okay. So stem cells are essential and defining feature of multicellular organisms, unlike archaea, viruses, and bacteria. Because you will study that when you start talking about architecture of the genome, the stem cells become, especially the embryonic stem cells, is where they have done a lot of work. Okay, because as I told you, these embryonic stem cells have 95% nuclear material in the cell, and there's only 5% cytoplasm. So So what happened? You are able to see the distribution of various chromosomes, okay, even in the interface. So that becomes this one, because once it is a adult cell, it is already defined. So it's very difficult to study the genome organization there. So as a concept, stem cells have similarities and differences in both animal and plant kingdom. And I told you it is unique to multicellular organism. The way both systems organize their stem cell pool suggests that there might have been a strong evolutionary constraint that shaped the path of development of this thing. Because you will soon, I'll be showing you some nice experiments, which will this thing, okay? So as I told you earlier, totipotency is, I told you, in case of a mouse, you after fertilizer divides into two, you separate the two, put it into two surrogate mothers, it will produce identical. You take 4 eggs, put it into 4 different surrogate mothers, it will produce this. But the moment it becomes 8, it doesn't do it. So it has lost the ability to totipotency in this one. In plant system, it's very easy, so that's why plant tissue culture is possible. So I want you to understand that. So totipotency is this thing, but otherwise what we call is pluripotency. That is, it can make only go into certain pathways, which I'll just show you. Now, I also want you to understand that Louis Pasteur was the first one who showed that theory of spontaneous generation doesn't happen in food exposure. So, a new cell is made from an old cell by division, okay? And that's what his classic experiment is, which all of you should read. In multicellular organ, be a plant or animal, spontaneous regeneration throughout life of a certain cell, tissue, organ, or whole this thing, it's very common, okay? And I'll talk more about it, at least one of the systems I'll try to do, okay? Self-organizing are an internal process, not depend on external, which I have been repeatedly telling you. Unlike animals, plants have the dramatic plasticity to take on multiple fates during their life. So plant system is quite different from the animal system, which you should know. Okay. Now, just to give you a background, so as I told you that there are about 10 to the power 14 cells in an average human being, okay? And assuming there are 10 to the power 14 cells, the number of cells types are only about 218. These 218 cells are not made by 218 mother cells. But what they found, the number of cells from which this might be created might be hardly 30 or 40. And one of the most well studied is what we call as blood cell formation. Okay. And as you can see, this mother stem cell, about which I'll talk now, one can create both what we call as your myeloid progenitors and lymphoid, your innate immunity, etc., all comes from this mother one. Okay. So as you can see, it can produce megakaryocytes, it can produce thrombocytes, erythrocytes, neutrophils, basophils, eosinophils, monocytes, a whole lot of things are T lymphocytes, B lymphocytes. So hematopoiesis is a very, very hot area, and I did quite a bit of work on that in the early years, and I'll just share with you what I'm going to talk now. So, so I said there are about 30 to probably maximum 40 stem cells, and for each process, like skin stem cell is there, hematopoietic stem cell is there, but as far as pancreas is concerned, we still don't know whether there's a stem cell, okay? So those days, I was interested in a molecule called ubiquitin, because ubiquitin had become very famous because the proteins have a shelf life. So proteins are created, they do their function, they should be destroyed. And the destruction mechanism was shown to be ATP dependent, and ubiquitin was involved. That was the very classical. So there is always a turnover which is happening. For example, say look at this, when the sugar level goes up, insulin has to be released. It breaks down, it helps in the transport of insulin from into the liver cells where it is metabolized. And the moment the sugar level goes down, insulin should be destroyed. Otherwise, it can create a havoc in the system. So this is what we call as intracellular role for ubiquitin. So one of my interests was whether ubiquitin has an extracellular role, and I looked at the hematopoiesis as a system. So I did a lot of experiments. I won't go into the details of it. What we found was that the mother stem cell, from which all these cells, T cells, B cells, can be stained without antibody using ubiquitin, and we showed that there is a ubiquitin binding pocket on the mother stem cell. Okay, these are also called the transit amplifying cells. So what happens is that whenever a stem cell, mother stem cell divides, one remains a mother cell and the other one differentiates. And you can see the red color there. This was stained with streptavidin, biotin streptavidin alkaline phosphatase system. You will hear that being used for many other experiments today. But I'm talking of this work we did somewhere around late 80s, okay, in India. Okay, it is not a foreign work. And what we were amazed to see was that there are only 95% nuclear material in the mother stem cell and only 5% cytoplasm. And we found that ubiquitin is responsible for the homing and adhesion of bone marrow cells. Now, just to digress a little bit, one of the ways to solve a lot of genetic diseases, for example, somebody has thalassemia or somebody has sickle cell anemia, if you have a matched donor, which is HLA match, you can take a normal one, destroy the patient's thalassemic cells or sickle cell, and then transplant it. Transplantation, nothing but you inject these cells. They will travel home and adhere to the bone marrow and started the whole hematopoiesis. So this is what is called bone marrow transplantation. See, today you are talking about genome engineering and all that. It has to go through a lot of clinical trials, but this was developed by Donald Thompson, HLA typing. And so one of the ways, if you have a ready-made cell available, which is perfectly normal, you can remove the mother stem cell, which is producing the defective one. Same thing we talk about cancer also. Cancer is also due to cancer stem cells. If we can remove that and put a normal cell into mother stem cell into it, it will proliferate and keep on producing normal cells. So that's how you find that leukemia and all are treated by this method. Okay, it's a well-established method. Okay. Now the very interesting thing what happened was, we said it is an irreversible process till Shinya Yamanaka in 2005, 2006 showed that if he put some 4 factors, OCT4, SOX2 and all that, he could make it become an embryonic stem cell. Okay, and there is a lot of excitement because now you could convert an epithelial cell back into the original one. Then you can differentiate it into whatever it wants. And this is called induced pluripotent stem cell. Okay, a lot of exciting work is going on. So this area is going to be competitive with genome engineering. Let me tell you that. Okay, so just to cut a long story, now I'll give you 2 minutes break if you want to ask anything before I go into the genome of eukaryotes. Any questions or any question in the chat box, Utpal?
So we'll take a small break as you are suggesting, and some participant also I also suggest. Sir, it's a kind request. Kindly make available all this session in the YouTube for future reference. Yeah, that will be done.
Okay.
This is by Tapashri Shur. So we are already recording it and it will be done. I was late in joining by 15 minutes. That's fine. So, sir, my suggestion is we may take a small break because the learning curve goes down if we continue.
Yes, that's why I said I will do.
Yeah, so that's fine. And actually 2 hours is quite a long duration. So we can take 5 minutes break.
Yeah. And or if somebody wants to ask any question, they can ask.
Now what we'll do, sir, I think a biological break will be helpful to all.
Okay.
And we'll start the session with 1 or 2 questions or few questions. So I request participants to post the questions and take a biological break of 5 minutes, and we make it very, friendly, everything. Yeah. So there is a question on Darwin's view on germplasm theory, how it gets related. So you can be ready. You need not answer right now, sir. I'm just telling you. You can also take a break, sir. And,
No, I'm okay. I'm fine.
unfortunately, the good thing about having workshop in physical presence is that we can provide the hospitality. Now you have to help yourself at your home. Probably madam will not be much irritated. Right. So Sonali Kalkar asking about stem cells in plants. Okay, that's very interesting. We will take up this question. Currently, we have two questions. One question regarding Darwin's view on germplasm theory, how it gets related, and Sonali Kalkar, stem cells in plants. Yeah.
Okay.
Uh,
So Darwin's theory, Darwin said there is evolution, okay, but he did not know what was evolving, okay. So then people related that it has to do with the genetic material, Mendelian factors, okay. And the classic experiment actually happened with the work of T.H. Morgan, okay. He was working on Drosophila and he found that he found some in Drosophila. In the female one, there was a white eye produced during various crossings. And through a beautiful series of studies, he showed that the female are recognized by the XX chromosome, whereas the males are recognized by the XY chromosomes. So if the male's X chromosome will be the carrier, so if both the affected chromosomes are there for the white eye in the female XX, then only the white eye is produced. That was the first time it was clearly established that the disease is caused by the change in the mutation of the X chromosome. Okay. So Darwin only said evolution is happening. Now this white eye was the one which actually gave us an idea that if there's a change in the base in the X chromosome, that can change. Now what that change and all that we came to know much later. For example, if you look at what is called sickle cell disease, first time we discovered that it is a change in amino acid. And amino acids are so a 3-letter code is there for each amino acid. So later in 1958, we came to know that the hemoglobin gene is affected, and when there is a gene mutation, it leads to a change in the amino acid. And so these are all later developments. But now we are talking of Darwin, Mendel in the 19th century, when none of these concepts were there. So slowly we could link the Darwinian concept with the Mendelian concept and what is responsible for the change. Have I answered your question?
Sir, she will respond. I am looking into the chat box.
Yeah.
So there is another question. Sir, please explain stem cells in plants by Sonali Kelkar. Maybe, sir, you can be very brief because we have 3 or 4 more questions.
Yeah. So
we'll go to
basically, your plant tissue culture is only happening because of the stem cells. Okay. What you do in plant tissue culture is you catch the callus, which is rich in. So there will be some stem cells there, which is what is giving rise to that. Okay.
Yes. So now, sir, there is another question. I will go serially, sir. What are the best softwares for biomedical human extraction? So she's asking about some softwares which
we'll discuss. We'll come to it a bit later. Yeah.
Brief question, sir. This is an interesting question. Can cell differentiation be reversed? Samrat Bora.
That's what Shinya Emanaka did.
Yeah, the Japanese.
Japanese by 2005. That is called iPSL, induced pluripotent stem cells.
I had the fortune to actually visit that cell repository where these cells are still kept alive.
Oh, lovely.
Yeah. So what is the recent? Another question by DC Bhattacharya, sir. What is the recent development with tardigrades? If cell cycle is known in details, I am not sure about this. Something.
Something is missing. Some word.
Yeah. So maybe you can explain the question a little bit more. Then another question is, as formation of an organ required interaction of many specialized cells during embryogenesis, will we be ever create such an environment in vitro to self-assemble a whole organ? I think, yeah, this is being done. I mean, in certain cases for skin tissue engineering, and actually the tissue engineers are working largely on this area.
They are only picking up the stem cells and putting them and putting the
Yes,
growth
yes,
factors,
yes.
and it's happening.
Oh yeah, so DC Bhattacharya is telling tardigrades are considered immortal in recent publications. I am also not aware of this development.
Okay.
Yeah,
I'll
maybe
pick up and get back to you.
later,
I don't know
later.
much
Yes,
about
yes.
it. Yeah.
So another question by Geeta Bharti. There are a lot of questions. So this was a small break. We can come back to these questions at the end of the session also.
Yeah,
But maybe, sir, we
sure.
can take one or two questions. Geeta Bharti is asking, in the process of survival, what monitors the success of some and the extinction of others? Okay.
Okay.
I mean,
Yeah, well, it is like, for example, you have the classic case of COVID now. Everybody is talking about Delta variant is able to expand much more than alpha or beta. Okay. So same thing happens in this thing, that if somebody who is better suited to grow will survive better than the one earlier one.
okay.
So I
Another
think
question
COVID is a
is,
beautiful example. Yeah.
another question is, how far is in silico methods applicable in stem cell technology? In
Well,
silico
people,
method.
yeah, in silico, basically what they are doing is, whether it's a stochastic process or deterministic process. Okay, that's a whole mathematical game being studied and trying to be stimulated. Okay, so
Yeah.
that's a whole mathematics of doing stochastic process and deterministic process, because stem cells, whether it is stochastic process or deterministic process or both, has been a question a lot of people have asked. When does it become deterministic? For example, if you look at that mother hematopoietic stem cell, at one stage it could have gone into the myeloid, this thing, or it could have gone into lymphoid. But once it becomes myeloid, lymphoid, it becomes deterministic that I'll only make these kind of cells. Lymphoid will only make those kinds. So at some point, the stochastic, some point it determined. People are trying to do modeling for that. Mathematical modeling.
There is one, this is last question we will take. Sir, just like the research is going on in developing a live planaria from its stem cells, can we culture other complex organisms from their stem cells? Probably he is telling, can we develop? Can we
We
develop?
can. In principle, we should be able to develop, but I think we are quite far from that kind of engineering yet. But it will happen.
Yeah. Yes,
Okay.
yes. Okay. Okay,
Yeah.
I think
I'll continue now.
people
Okay.
have come back around 103 attendees now. So there are
Okay.
one or two questions, so which we'll take up later. Let us go ahead with the main session. Yeah, thank you.
Okay. Okay.
Yes,
So
sir.
now I am coming to our modern version of the genome. Okay. So genome of eukaryotic. So this is a standard one which all of you would have read. And I was very fortunate to, know Professor Aaron Klook, who did most of this basic work, the nucleosome, zinc finger regions and all that. So as you can see, a eukaryotic genome is organized in the form of a highly complex this thing called nucleosome, the basic unit of chromatin, consisting of 147 base pairs of DNA, wrapped 1.65 times around the histone octamer, composed of core histone H2A, H2B, all of them occur in 2,2. So H2A, H2B, H3, and H4. And interestingly, ubiquitin is attached to H2A and is called a A24 protein. So originally, ubiquitin was discovered as a A24 protein way back in 1975. Within the chromatin, the distance between two consecutive nucleosome core varies between 20 to 100 base pairs. So we are looking at the micro level, how the genome is organized. Okay. Base pair of linker DNA that associate with a special class of histone known as the linker histone. Interestingly, these work were all done by X-ray crystallography. And you must read the R.N. Klug's work, because he was one of the ones who promoted, and many in his group went on, and some of them went on to win Nobel Prize also. So this nucleosome is essentially a complex of protein, and people have worked this out, how many H2As are there, H2B, H3, H4, and what is linked to that. These are all done with what is called, which we will come to know, that there are some chemical reagents which can interact and lock them up. You take it out and see which I'll talk in a few minutes from now. Okay. So nucleosome was the first one which was thoroughly studied. Okay. And then several nucleosomes get organized on chromosomal DNA in a linear beads on a string pattern connected by the linker DNA that constitute a nucleosome array, forming the lowest functional unit of chromatin compaction and is permissive to transcription. So this is what our current knowledge is. Further, organization in the form of 30 nanometer chromatin fiber is thought to be repressive to transcription events. So what happened, this is not a linear structure, but it folds into, just like protein folds into alpha, beta, but here it is much more complex, because one whole human chromosome, if you put it, it will be about 2 meters long. That's about 6 feet long. Whereas our cell is so small, so the whole thing is formed, and that is what is called the linking number paradox. Okay, that is, how is this linkage happening? And these are all the questions which used to be asked in the 80s. I used to hear Sir R.N. Klug speaking on linking paradox, because Crick and R.N. Klug and all used to mathematically question a whole lot of issues which happen. Because even now we say which part of the genome is accessible for transcription and which is not. So I'm just giving you an idea that that the organization into 30 nanometer chromatin fiber is thought to be repressive to transcription event. Another level of compaction is brought about through the histone tail mediated interaction of 30 nanometer chromatin fibers. Formation of chromatin thus adds hierarchical compaction compartmentalization. We'll go into, so this is the current information which is available. Despite the high degree of compaction, DNA should be, I don't know, to snap it. Okay. Should still be readily accessible to allow interaction with several different protein factors, regulating its functions such as replication, transcription, repair, and recombination. The continuous interaction of chromatin with a myriad of proteins, including epigenetic factors, chromatin remodelers, and histone chaperone, which act either in an ATP-dependent or ATP-independent manner, is vital to maintaining its dynamic nature. The dynamic nature of chromatin thus influences almost all genome function. Recent years have witnessed rapid progress in the structure and function of chromatin. However, these studies were mainly focused on a few biological problems. Okay. So now let us look at why we are interested in studying engineering the 3D genome, because there should be some reason why I should do engineering. Because I told you earlier, I can do a stem cell transplantation and cure many diseases. But we have not been successful because it's a very long drawn process. I'm sure when we do engineering also, we'll have to do a lot of clinical trials. And what are the, because we have to remove the defective cells and remove it. So probably any experiment you finally gets into the translational science, it has to go through the similar process, bone marrow transplant, in the sense, you know, it takes about 6 weeks before it gets transplanted. Okay. So people have found that cancers and developmental disorders are associated with alteration in the 3-dimensional genome architecture in space and time. So that is the first thing people observe, that if there is a conformational change, that also can, just like if a protein gets denatured, it is no more, enzyme is no more active. So the structure is very inherent, and how stable is it? So actually I can do a comparison between protein studies and genome studies. Both are similar in the sense that approaches are very similar. Chemistry is similar. Okay. So as we go along, I'll explain to you what I mean by this. Okay. Mammalian 3D genome organization is complex and dynamic and plays an essential role in regulating gene expression and cellular function. Okay. And that's what we are trying to address now. To study the causal relationship between genome function and its spatio-temporal organization in the nucleus, new technologies for engineering and manipulating the 3D organization of the genome have been developed. Okay. So one of them, today we are very lucky because CRISPR-Cas gave a big boost to this field. Not that other methods were not known, but the progress of work was very, very less. Because of Doudna and all that, her work and all that, CRISPR-Cas technology became one of the biggest thing. And today we are expecting a whole lot of new results which is coming out. Okay. Let me be very honest. Now I'll be talking basically based on the review by Haifang Wang, Menking, and Lee Sequei to the very recent review. So I'm going to cover most of the things now out of this work. Okay. So just to give you an idea, you have in protein, say we have primary structure. So people like Sanger and others develop methods to sequence Edman and all this. Okay, one is primary. Then you have what is called secondary structure. Then you have tertiary structure, which is determined by X-ray structure. So for each one, you need a different kind of technologies and techniques. Okay. And today, of course, technologies have advanced much more, but I'm just giving it. So similarly, the question you will ask is, why can't I study 3D genome? So first, we did not know the sequencing of this, even though Sanger's method and all didn't give. But today, with the next generation sequencing, it is possible now with the Oxford nanopore technology and all, which does sequencing of 400 base pairs, 400 bases every second. That's why you hear the COVID sequences are done in an hour, because the rate at which the machine does is so far that you can find out. So somebody lands in airport, you're able to pick up and do. So you have one question of a rapid way to sequence. Okay. So, and the technology is totally different because a micro level of the thing, and it's a nanopore through which it goes. So you get the primary structure, but that's not enough to explain the phenomena. Okay. So you need to know the 3D structure because biological functions happen on 3-dimensional level. So people have looked at, many reviews have been written. Okay. So from including sequence. So in all these things, you will see that finally you get the DNA interacting with another DNA, or a DNA interacting with a protein. You cut it with a restriction enzyme, just like we used to use proteolytic enzyme to do the sequencing. We use restriction enzyme to cut it, ligate it, then do the sequencing. Okay, and find out what is the sequence of the DNA, and then what is the protein bound to it. Okay, so one of the very popular one is what is called chromosome conformation capture. That is called 3C technology, which was developed around 2002. Now there are a lot of the 3C, 4C, 5C, high C, a lot of modification. Since others are going to talk in great length about it, I just want you to know that to catch the chromosome conformation capture, one of the very popular methods is to use formaldehyde. So formaldehyde will cross-link 2 chains. Suppose there's a loop, and it will link which is next to which one by forming a cross-link. So formaldehyde. Now the chemistry of formaldehyde is quite a complex one, because you should know good chemistry. Okay, that when you get it, is it valid? How do you do a positive control? How do you do a negative control? What is the difference between in vivo experiment and in vitro experiment? The linking might have gone somewhere else also. How much to use formaldehyde? It's a subject in itself. So 3C technology, talking about the experiment itself, is quite complex. And it will generate so many fragments that to analyze them, you need a software. So that's a different ballgame. Another one is called DAM ID. That is, this is what we call a DNA adenine methyl transferase. Especially if you look at the nucleus is a porous one. So these are called laminins. So whole set of chromosomes which are inactive, what we call as heterochromatin, are very close to the nuclear pore. So to identify the proteins which are interacting with the heterochromatin, the DAM ID, that is what I said as DNA adenine methyl transferase identification, is a technique which is used. Okay. Another very popular one is called SPRITE, that is split pool recognition of interaction by tag extension. This is done on microwell plate. So there also it creates a whole large number of data. Okay. Then you have what is called genome architecture mapping, what is called GAM. Then you have chromatin, immunoprecipitation. Okay. And there are a whole lot of variation of that, what is called CHIA PET. Then with this nanofluidics coming, there is what is called drop method. Okay. So the whole lot of techniques have been given. I'm sure many of your speakers are going to speak details of this. Then another area which becomes very important is imaging. And the most popular I remember is I used to do a lot of fluorescence in situ hybridization, especially to find out chronic myelogenous leukemia and all. Because earlier, before FISH came into picture, we had to always do karyotyping. And people used to do the karyotyping and find out what could be the genetic cause of the disease, like chronic myelogenous 922 translocation and all that were detected by studying the karyotype. But for that, you needed metaphase. But with the FISH coming, you didn't need a metaphase chromosome. You could directly take at the interphase and stain it, and you will find that it shows up the fluorescence. Because if there's a translocation, then you can have, say, a green fluorescence for one part, a red fluorescence for another, and if both of them have joined together, then you will get a yellow color. So that is the standard way fluorescence in situ hybridization work. And now anything you attach a fluorescent probe, you can do a live cell imaging, okay, regarding nuclear organ chromatin conformation, okay. But ultimately, 3C technique is very important to confirm. Of course, today with cryo-electron microscopy, you can have the frozen sample and make section, take out the DNA and look at it also. And with the cryo-electron microscopy, you have an overall picture, so you can have segments. So technology has gone at a different level altogether. And remember that because of technological advances, many techniques which are common have become obsolete or becoming obsolete. I want you to have a look at, go yourself and search what is the popularity of 3C technique, what is the popularity of DAM ID, or is it falling down, or SPRITE or GAM. So you will find that with the advancement in technology and microfabrication, etc., many techniques which are important 10 years back might be losing its significance. Just like I was a protein sequencing person in the 70s. Then, with the DNA sequencing of Sanger came, you could arrive at the protein sequence directly by reading the DNA. Now what we find that now protein sequencing has again become very popular because why it has become popular? Because now I am able to analyze 600 molecules within a cell. Earlier, if you remember, like for example, your spectrophotometer and all will need anywhere between 10 to the power 16 molecules to see. But today with the fluorescence, people have gone what is called zeptomole sequencing. Zepto means 10 raised to minus 21 moles. So technology, fluorosequencing will change. So protein chemistry is going to come back because DNA gives you this thing, but a gene expression will show you something, but you don't know what are the products formed. Whether it is a post-translational modification, what is happening, what is the concentration, all this is today possible due to fluorescence. So fluorescence has become the mainstay. Okay, although these mapping approaches are powerful at depicting how 3D genome organization changes across different differentiation and disease stages, they mostly provide correlated information on the 3D genome function. To better understand the causal relation between 3D genome organization and cellular function, it is important to develop manipulative techniques to this thing. That is why you are trying to change a base through CRISPR or TALEN or whatever, which we'll come to now. And then see if I change this base, what happens? Will it create a disease? So it will have disease models which will come up. Okay. So this sudden excitement has only happened because of CRISPR technology, CRISPR-Cas technology. So that is why you will suddenly see a powerful system for genome editing, because you are able to change a base. So you can do a lot of things now. Gene regulation, epigenome editing, 3D genome engineered control spatial and temperature, etc. So there are a lot of excitement today. Okay. Now, before this thing came, there were other techniques, as I told you. So one of them are used to build eukaryotic toolkit. One is sequence-specific gene editing system, such as zinc finger nucleases, about which I'm sure you're having a separate session. You have talents, you have another session I saw in your program. You have CRISPR-Cas. Then using the non-nuclear sequence-specific DNA binding system, the LAC apparator, LAC repressor, zinc finger, transcription activator, a whole lot of things have been this thing. And I don't want to go into detail, but I want you to know that the many speakers are going to speak of this in great detail. Okay. So zinc finger nucleases are all artificial protein consisting of an array of zinc finger DNA binding domain fused with a non-specific DNA cleavage domain from the restriction enzyme. Okay. Anyway, the details will be talked to it, so I won't go into details of it, but wait for the main speakers who will speak with. These are all subjects in itself. Okay. So this is one of the typical ones. So LACO, the artificial array containing repeats of bacterial LACO sequence. Now you can design all this thanks to, genetic engineering, and design whatever you want, which will bind there, etc. You will have more, you will hear from the other speakers. Okay. And so now you have to deliver this one, and then how the whole editing happen is done. So you need some nanoparticle to deliver it. Luckily, the nucleus is porous, so it can go through it and then do whatever the repair it has to do. Okay. And this is the typical zinc finger design. This work was essentially done by Aaron Klug and his group, whom I had the pleasure of knowing. And he was the man who started talking of this in great details. Okay. So you can see what happens in zinc finger. And then this is another very popular. There are so many systems. I can't cover all of them, but I just thought I'll help you to be familiar with, because you can LAC operon, so it's very well known, so you can use it to study this thing. Okay. Then there are a few terms which comes, what is called chromosome territory. So we are talking about interface, and I told you many of these studies have been done with embryonic stem cells. And I said all of them have large nucleus, 95% nuclear metal and very little cytoplasm, 5%. So it becomes a whole canvas. Okay. So most of the study you will see is that. So in that, when you look at it, there's a chromosome territory, the distinct volume of nuclear space occupied by each chromosome. Then, okay. So now there are so many techniques I told you. I don't want to go into details of it, but these are all ways to edit the genome. And just leave it there, because you will have detailed lectures by experts in each one of them. Okay. And I was telling you about this chromosome conformation capture technique. So a technique, I told you they use formaldehyde, but it's quite a messy thing to sit and analyze. Okay. So you have to be a good nucleic acid chemist, a good protein chemist to really understand what are the ones which are nearby, which are not nearby, all that thing. You can literally map this thing. So I'll just come to, there is what's called heterochromatin. They don't express, they are quiet. Whereas you have euchromatin. So you can see in euchromatin really what happened. by this 3C technique. It's hardly 20 years old, okay? And there are a lot of variations of it, so the literature is full of them. You can keep talking in great length, but the point is you have to understand how these techniques are used to understand the three-dimensional structure. So remember, it is the understanding of the structure, how you interpret is your brilliance, okay? And what kind of software you write, and whether it's a positive control, negative, how do you know this interpretation is correct or not? So you'll have to go back and actually check in the experiment. Whatever the software reads, you must have an independent check and balance on this. That's why it's quite a laborious work, okay? Fluorescent in situ hybridization, many of the probes have at least minimum 30-40 bases there, and that is used to, and you label it with some fluorescent probe with that, then it will specifically say where that sequence exists, that long sequence. Then you have what is called CRISPR live cell imaging also you can do. So you put a tag, you can literally see what is happening. So heterochromatic, these are all terms you should be familiar with if you want to understand genome architecture. The condensed region of a chromosome that is tightly packaged, which is usually gene poor. Okay. Heterochromatin contains repressive epigenetic markers and is less accessible to transcription factors and transcription machine than is euchromatin. And normally this heterochromatin is found on the very close to the nuclear membrane. Okay, and the active euchromatin is normally found on the center. That's what people have this thing. So the less condensed region of chromatin that is lightly packed. So you will see like chromosome 15, 16, 17, 18 will be missing because 18 doesn't have much gene. That will be more in the heterochromatin. 19 does. So people have seen that these genes sit together in the center of the nucleus. And they are accessible for transcription factors than is heterochromatin. Okay. So hierarchical organizing a 3D genome in the nucleus. At the nuclear organization level, each interface chromosome occupies distinct region called a chromosome territory. So it is just like nations have boundaries, chromosome also have a territory. Okay. And membrane-less nuclear components such as nuclear bodies, nucleolus also interact with them. Okay. So it's quite a complex phenomenon. Radial positioning of the chromosome and genomic regions in the nucleus is associated with their gene activity, with inactive regions located close to the nuclear lamina. I told you near the nuclear membrane. Okay, forming lamina-associated domain. It's a very popular word you will hear during others' lectures called LATs. According to a loop extrusion model, loop extruding factors like cohesin actively extrude loops until they reach the boundary. Okay, one is CTCF binding proteins. Okay, specific loop interaction can be stabilized by structural proteins such as CTCF, cohesin, and yin yang. So these are all words you will keep hearing in the next 4 to 5 days. At the linear chromatin level, chromatin nucleosome fibers are formed by the linear DNAs assembling with histone, which collectively harbor various DNA modification, histone modification that are involved in epigenetic regulation. Additionally, different DNA motifs can be recognized by structural proteins and transcription factors for genome organization regulation. Collectively, these different layers of genome organize, regulate gene expression, subsequent function. At the chromatin organization level, chromatin is segregated into regions that assemble into A and B compartments. A compartment generally harbor active euchromatin and tend to concentrate at the nuclear interior. I told you it comes more in the center, not in the periphery of the nucleus. B compartment usually harbor inactive heterochromatin and prefer to locate at the nuclear periphery. I told you there. So B compartment is normally seen near the periphery, whereas A compartment is seen at the interior of the node in the somewhere around the central region. Local chromatin forms sub-megabases, self-interacting domains called topologically associated domains. Okay. Now, just to give you an idea, many times we ask, everybody is today talking about antibodies. What do antibodies see? in a protein or any other system? Is it seeing a primary structure or is it seeing a secondary structure? Okay, so these are the questions. So now he is now for the first time they're talking about topologically associating domains. Okay, so to probe that you need antibodies to look at that domain. Okay, so this is becomes very, very important. Okay, then there is what is called nuclear bodies, a collection of membrane-less structures in the nucleus with diverse cellular functions. Different nuclear bodies are enriched in different structural proteins and RNA components. So it's quite a complex system, the nucleus. There's a lot of nuclear bodies there. And lamina associated domain, the regions of chromatin that interact with nuclear lamina at the nuclear periphery. They usually contain inactive genes and can be mapped by sequencing approach that I told you about DNA methyltransferase, okay, DAM identification. Then there are topological, so these are all terms you should be familiar with, okay, if you want to understand, because these terms will come repeatedly, so you should be aware. So topologically associated domains are one, local self-interacting chromatin region that are often separated by boundary elements. DNA elements exhibit high frequency of contacts with regions within the same topologically associated domain than interaction among topologically associated domains. Then there is what is called a loop. So please remember, these are all words you should be familiar with, what they stand for. So as a teacher, when you teach, you'll have to explain all that. A DNA loop is formed when two genome regions in the same chromosome contact, which can bring two distal DNA sites together. And if you do a formaldehyde treatment, they'll be logged. So you can pick it up and do that. Mediating interaction between cis-regulatory elements such as promoters, enhancers, insulators, and silencers. The insulator is a very interesting one because insulator one means that gene won't be expressed. For example, say you take the mother cell and say, you're talking of T cell, B cell. Each one expresses certain genes only. So, and all of them have the whole chromosome. All these cells have the whole chromosome. But with the insulator, they are prevented from expressing. So each cell has a characteristic genes which it will express. That's why we call them as biomarkers. So we might look for a product which it makes, which is unique to that cell. So in other words, when the differentiation happens from the mother stem cell to become an adult cell, certain genes are switched off. Not that they disappear, they're all there, but they're all shut down. Okay, so that is the fundamental thing you have to understand, that how these switching on, switching off happen. What is the role of the promoters? What is the role of enhancers, insulators, and silencers? Okay. And CTCF binding, a conserved 11 zinc finger protein encoded by the CTCF gene that binds to a conserved DNA sequence, such a long one, CCG, CGN, GGN, GGCAG. Okay. Cohesin complex is there's the one which makes the loop, protrudes the loop. A protein complex composed of 4 core subunits. So each subject, each definition is a complex in itself. Okay, so all this has to be understood to appreciate the 3D structure. A protein complex composed of 4 core subunits, SMC1, SMC3, RAD21, SA1, SA8 can form a ring-shaped structure enclosing DNA and plays important roles in mitotic and interphase chromosome organization. Then you have CTCF binding site, what is called CBSS. The binding sites are CCCT binding in the genome that can contribute to organization of topologically associated domains and loops. Insulators, cis-regulatory DNA that block inappropriate interaction between genomic regions. Then you have what is called super loops. Network of super large loop that exist on the inactive X chromosome around the DXZ4 locus. So just to give you an idea, I'm just taking one example. Okay, there are so many methods, so I'm talking about zinc finger nucleus deletion. Targeted domain, we just talked about CBS, okay, and target gene is so and so, okay. Then alteration disrupts original chromatin interaction among insulators and enhancers. Cell type used is NT2 or D1 human lung carcinoma. Then implication is related to retinoic acid signaling control chromatin modeling. So genome editing, to study 3D genome, you can do edit the structural DNA motif, manipulate DNA looping, editing structural proteins, engineering nuclear repositioning, genomic repositioning by tethering a compartment-specific protein, genomic repositioning by recruiting transcription and chromatin. So there are so many ways to do it. So it's quite a complex subject, and I'm sure the rest of the speakers will talk about some of them in great detail with a specific example. Okay. So manipulating nuclear DNA interaction. The eukaryotic nucleus contains abundant membraneless nuclear bodies that are functionally important in a variety of biological processes. In fact, the first one was discovered by Kajal way back in 1902, called Kajal bodies. Then you have nucleoli. Then you have PML bodies, promyelocytic leukemia bodies, nuclear speckless and para. So these are all things seen inside the nucleus. For example, Kajal body often associated with small nuclear RNA, and histone gene harbor centers for biogenesis of small nuclear RNAs and assembly of ribonucleoprotein. Some nuclear bodies are proposed to assemble through liquid-liquid transition driven by multivalent interaction between structural proteins and RNA. So, today we are talking so many types of RNA, and they're all coming from there. So now we are understanding much more in depth from where they come from. Synthetic approaches have been developed to modulate the interaction between chromatin and nuclear bodies and to study the dynamic mechanisms and function of nuclear body formation. We are going to hear from some of the speakers who will talk on this aspect, okay? So tools for engineering chromatin nuclear bodies is, we talked about the induction of chromatin nuclear bodies by recruitment of structural proteins via LAC-I system in a stable cell line containing an array of LAC-O repeats, recruitment of NB structural protein, etc. This is the whole, de novo formation of nuclear bodies, for example, Kajal bodies, etc. So you can do a lot of these kind of engineering you can do. Or another induction of chromatin NB by recruiting RNA components. Okay, that's what is talked about in great detail. Third is induction of chromatin nuclear body via CRISPR genome organizers. So, or alternatively, lengthening of telomeres, etc. The whole lot. So it's tools are so many for engineering chromatin nuclear body interaction. Engineering hubs. Now, there is another thing, that what is inside the nucleus, that is another very interesting. What is the liquid-liquid phase separation, which is separating them to keep them in different. There must be some density gradient or whatever it is. So many nuclear compartment body proteins, RNA binding proteins are contained intrinsically disordered. Now, this is a very important subject, what is called intrinsically disordered regions, okay, or low complexity that can interact with each other and form phase separated condensates in vitro. Just to give you a simple example, if you see milk, which all of you are familiar with, milk is made up of, you have caseins, you have whey proteins, etc. Now, the milk is white due to the formation of micelles, okay? So these are protein micelles. And if you analyze a protein, there are about 18 different proteins in milk. So the fastest one is called alpha, the other one is called beta, gamma, kappa, etc. The one which is interacting with all of them is a protein called kappa casein, okay, which is very interesting in the sense that it is a 1/3 hydrophilic and 2/3 hydrophobic. It's intrinsically disordered. but the moment it comes across alpha, beta, gamma, and all that, they interact and form a very beautiful micelle. And the white color of milk is due to the scattering by these micelles. It can be 100 nanometer particle, it could be 500 micrometer, depending upon kind of sugar which is present on the kappa casein. Okay, I won't go into this. So these are called intrinsically disordered proteins. So even your transcription factors are all intrinsically disordered. So by seeing a structure in interaction, it forms an ordered one. But if you were to isolate them and study, you can't even make out how they are interacting. So it is a localized effect which happens, which brings about the right fellows meet, the whole structure develops, it evolves. That's why we call it an intrinsic mechanism. You can't control it. So what you put, so there are so many transcription factors. You might ask, how come they are going and so specific and doing that? Because they get ordered when they see only a particular, otherwise they will not get ordered. So that's so there's a whole area of research called intrinsically disordered region, intrinsically disordered domains, etc. Okay, so this is a typical one. So nuclear compartment of RNA binding intrinsically contain disordered regions, and how they interact is something we have to understand. It's proposed that LLPS provides a physical mechanism for organizing nuclear condensate. What is it? Liquid-liquid phase separation. Okay, provides a physical measurement for organizing nuclear condensate, including membraneless nuclear bodies, compartment, transcriptional factories, which can favor certain biological reactions. I told you, under certain substances only they will go and interact and form. Otherwise, they will not. By selectively partitioning and concentrating specific molecules within the condensate while excluding other molecules. Okay, so these interactions are also not that simple. It is not like A base pairs with T and G with C. It is much more complex than that. So tools for inducing interaction hub. So that is where you change the bases or you change the amino acid and see how the interaction is. So induction of low complexity domains interaction hub formation by recruitment of intrinsically disordered regions or a low complex domains via this thing in a stable cell line containing array of lag co-repeats, recruitment of intrinsically disordered regions or LCDs of transcription regulators via direct this thing, which may recruit other LCN polymerases, and small LCD are also formed. Okay, the light inducible CRISPR-based Cas drop system to NUK. So what I'm saying, there's a whole lot of physical chemistry. Okay, and sometimes some of them are using even optical blue light induction also. There's a whole field called optogenetics, which is coming up to understand by shining light what happens. The nuclei are also formed at non-targeted nucleus space to shape this thing. So now we come to 3D genome engineering. 3D genome engineering tools are useful for investigating the causal relation between 3D genome abnormalities and disease occurrence. A variety of developmental diseases and cancers are associated with alteration in 3D genome organization, such as disruption of topologically associated domain boundary, aberrant enhancer promoter interaction, disease-associated genetic variants that locate in non-coding regions can often be associated with disease-relevant genes through 3D genome interaction. Engineering tools such as CRISPR techniques allow target-specific manipulation of genome sequence for 3D organisms, which has recently been used to build multiple disease models and to dissect the molecular mechanism related to. Only thing, we have to make sure that that point only the change happens and nowhere else happens, and what is the consequence of that. So let me conclude my part of the talk and give you a perspective. First thing I want to tell you, you do any of this, show it in an animal model, it has to go through the full clinical trials, okay? The whole game is going to be as challenging as bone marrow transplantation. Okay, so it's not that it's going to come tomorrow. It is going to through its process. It may take another 10 to 15 years before a translational product is likely to come into the market. Okay, so be prepared for it, but you are in a very exciting phase where you can study certain organisms, what happens to their development or what disease it causes, and then go back and look at human system, whether similar thing is what is happening. So it's a very exciting area, but long way to go because the regulatory issues are going to be very tough. So let us conclude what we want to say. So genome engineering is becoming an instrumental tool for studying 3D genome. So it's more an academic exercise right now. Editing structural DNA motifs and structural proteins is used to dissect their roles in organizing the 3D genome. Induced DNA loops are formed at specific time with the help of cohesin to mediate enhancer promoter interaction and to study the role in transcriptional control. Manipulating genome positioning and DNA nuclear body interaction allows us to understand their importance in cellular function, which aids the study of nuclear body formation and dynamics. Synthetic methods to recruit intrinsically disordered region, low complex domain reveal their roles in regulating transcription and organizing nuclear architecture. Together, the development of 3D genome engineering tools substantially will facilitate our understanding of genome organization principle and the causal relation between 3D genome structure and function. Owing to the high complexity and dynamic nature of 3D genome organization, many tools are built on chemical or optogenetic inducible systems for precise temporal and spatial control of targeted manipulation. Current tools, for example, induced loop formation or genome repositioning, have been tested on only a limited number of genomic regions, and it is necessary to expand these studies to understand the generality and context dependency of existing conclusion by testing more genome targets. With CRISPR technology, it is feasible to design guided RNA sequence target different genomic regions of interest. However, the possible off-target effects of CRISPR system also need to be taken into account and carefully evaluated. So if it goes and binds somewhere else and creates some other mess, so it might become more complex than what you started with. The manipulative approaches can be integrated with mapping and imaging methods to simultaneously manipulate targeted region and study the consequential outcomes on organization, dynamics, and function of 3D gene. For example, perturbation of DNA loops or topology associated domain and compartment organization by targeted manipulation can be further studied by sequencing methods such as 3C based technology, about which we talked earlier. Alteration of DNA replication timing can be profiled by RepliSec. Changes in chromatin lamina interaction can be studied by the DAM-ID method. Moreover, imaging-based methods such as FISH and CRISPR live cell imaging are being used to investigate temporal and spatial organization of specific genome region and the relationship with different proteins, RNA components, and nuclear components. Development of combining manipulation and sequencing may will result in better tools to better profile causal disease. 3D genome engineering methods offer new tools for studying mechanism of genetic diseases. Comparative 3D genome mapping and imaging studies are useful to profile changes in chromatin organ during development. And mind you, ultimately, the development should not be affected. It should give a positive solution. The combination of genome-wide associated study data and 3D genome mapping data may uncover key regions of genome organizing element related to various disease variants. These studies can provide potential targets for using 3D genome engineering system to validate a causal relation with genetic variant, 3D genome abnormalities, and disease state. To date, CRISPR genome engineering tools are mostly used for studying disease relevance of topologically associated domains and enhancer promoter interaction. Further development are to precisely manipulate 3D genome structure and function on different levels will further contribute to disease understanding, research, and therapies. So thank you.
Thank you, sir.
Mm.
So, dear dear participants, if you have some queries, you can type the queries in the chat box. So, in the meantime, yeah, there is one question which is very general.
Yeah.
Sir, RBC do not have nucleus, but how does this cell survive for 120 days?
Okay. So basically what happened, RBC is only the final cell. It goes through for various stages called blast forming units of the erythroid, colony forming of the unit erythroid, etc. Now, luckily, in case of RBC, nucleus is not there. So it is only a bag of enzymes and proteins and other things. So half-life, what we talk, say like 120 days, how it is cleared is very important because each cell has a shelf life. Okay. So in case of RBC, 120 days it is destroyed, and then mother stem cell will produce more RBC. So it's a continuous. Every day, the body produces 1 billion RBC, 1 billion platelet, 1 million T cells, 1 million B cells per kilogram body weight. It's a continuous process. And this is happening because you have a mother stem cell. It divides into two. One remains a mother stem cell. The other one divides and produces all this. So there's a constant bank balance kept. Is that clear? So as far as the half-life issue is concerned, 120 days, some of them may have more. Some of them, like insulin and all, are destroyed the moment its function is over. Some hormones only act for a very short time. So half-life is a separate subject. Okay. I hope I answered your question.
Yes, sir, it's I think very well explained. As you're rightly telling, RBC is only the final stage that we are able to see, but actually it had all the information. That's why it had produced all the proteins.
Yeah, its function is only to move oxygen.
Yeah,
that's all.
Right. So that's because of the absence of the nucleus, it cannot renew itself or divide.
Yeah, but there's a mother stem cell which will produce
Yeah.
again.
Correct. So one question, how intrinsically disorder protein maintain interaction specificity with multiple ligands? This question doesn't actually fall under this lecture, Dr. Neeraj Singh. But anyway, it's a question of protein-protein interactions or protein nucleic acid interactions.
No, that is why I'm saying, suppose there are so many girls, there are so many boys.
Yeah.
One girl falls in love with another boy. Why only one? Why not many?
Correct.
Is it specific? All are intrinsically disordered.
Yes.
Suddenly they meet the right person and everything clicks. So that is what happens in transcription factors and all that.
Right. So are there any other questions? If there is any other question, yeah, there is a question, sir. What is your view on considering axolotl, AXOLOTL in stem cell extraction and its studies?
It's a good model system to study axolotl. See, there are so many model systems, only few are available right now. I'm sure there are much more interesting systems to look for.
Yeah.
For example, say tilapia. I was told tilapia skin is very good for burn injuries. You can put the tilapia skin, and if somebody has a loss, burn injuries of a higher order, 40%, 30%, you can put those skins. So there are a lot of interesting animal models which can be used also. You don't have to always look at human systems alone. Okay. So axolotl is a very interesting system to study.
So there is another
systems to study, just like zebrafish. Who thought zebrafish is going to become an animal models? So somebody looks at it and finds some advantage in particular case. That's all. Yeah.
There is another question, sir. What are the chances of such gene engineering products of being inherited?
Well, if it doesn't affect the genetic, the what we call as the one, it doesn't affect the sperm or egg, then it is okay. If it only affects the somatic cell, it's okay. So they normally work only on somatic cells. Okay.
There's a question by Sangeeta. She's asking recent trends of use of these techniques in diabetes cure. Probably she is asking about stem cell therapy in diabetes cure or genetic engineering in diabetes cure. I'm not sure. She's asking a very generalized question. Recent trends of use of these techniques in diabetes cure.
See, problem with diabetes is basically the role of insulin is to make the sugar enter the liver. If the sugar doesn't enter the liver, so we still don't understand the details mechanism of sugar entry into liver cell. So what is the role of insulin and how it affects the sugar entry is the question. If it doesn't enter the liver, it remains in the blood circulation. That's why it is diabetic. So we still don't understand the mechanism of that. And number two, people thought there might be a stem cell for beta cells. But there is a two schools. So some say there is no stem cell for it. Some say there is. So it's still controversial. There's no resolution on this yet. I think Utpal, you did some work on tissue engineering,
Yes, sir. No, not in this area. In the nerve tissue engineering area, sir.
Yeah.
Yes. So from Dr. Geeta, I think it is not a question. She is telling she is working in autoism lab. That's not a question. Probably she wanted to tell something. Yeah, so we have taken up almost all the questions. Yeah, that question on target dates. Target dates are considered immortal in recent publications. But I think some kind of literature review would be required and or what is the quality of those, because the simple formula for life is not to be immortal probably. But sir, you can have a very general philosophical, treatment of this question. Target dates are considered immortal in recent publications. So, but that's not a question. So just like the research is going and developing, no, that's not a question. Okay.
Yeah, it's not a question, it's just a general statement. See, we have to go a long way. Ultimately, whether we can have a transplantable cell, could be a normal cell or an engineered cell. And now people are talking because we are not getting enough HLA match donors. Because in India, we don't have donor banks, where we maintain all kind of. For example, you are seeing, kidney transplantation, people are struggling because match donor is not available.
Yeah. Dr. Geeta has a question, but I think we have a lecture lined up. She's asking how to avoid off-target effects of CRISPR system. I think this question Dr. Geeta put up when we have the session on CRISPR-Cas9 system. Yeah. Right. Yes.
Now, only I just want to conclude by saying the teachers who are attending, don't get carried away by all these technologies. Be pragmatic in seeing how much is translationable and how much is not, because these are all R&D stage only. Until it gets translated, which is going to take time. So it is good as an academic exercise, but how much of academic exercise will become reality, only time will tell. Am I right, Utpal?
Yes, absolutely. So
don't get carried away by that is the in thing.
Yeah, the last lecture in your platform, that amazing figure of roughly around 60,000 papers on COVID published in less than 1.5 years.
Yes.
There's a question being raised about the quality of the papers. And now, so even the review process has been questioned. So these are very important things about the quality of science being published. And so one more question is there. Dr. Neeraj Singh, why microbiome called a second genome? Okay, yeah, it's a very good question.
It's a very good question because we have more microbes around us than we as a population. Yeah, and so they always interact because they go to our gut. Gut microbiology is another big field, and a lot of diseases are being associated with that. So we have to understand gut microbiome also. Am I right?
Yes.
Well, yeah.
Absolutely. Because I think if you just go to our traditional food habits of taking card and other fermented food, so we are getting a lot of benefits from these
Yeah,
beneficial
probiotics. Yeah.
and we don't have really many of those genes, essential genes, which produces vitamins and other critical,
Yes.
constituents of food. So they are there to support us. So that's why we call them as a second genome. And probably
Correct.
this is there's a concept of pan genome, where organisms actually do not carry genes themselves, but they rely on the community genes.
Yes.
And
Yes.
very often they acquire those through horizontal gene transfer. And also
Yes.
they get rid of them whenever they feel carrying those genes is an extra load. So it's more
Yes.
about, those are very interesting aspects. I mean, how do you economize, because particularly the bacteria are very small, and carrying too many genes may be actually not
Yes.
very viable for them. So a few questions again cropping up.
Yeah.
One of the reason proposed for autism disorder is gut microbiome, sir. Yeah, I think you are correct, Dr. Neeraj Singh. Now, actually, a lot of focus on the gut microbiome.
Yes.
In fact, including mental health, the gut
Yes.
microbiome has been held responsible. So there is now many studies going on various diseases and the connectivity of gut microbiome. And as you are telling, autism disorder, I mean, some kind of correlation people might have found out. But sir, if you'd like to add something on this.
No, I mean, I think a lot of research, these are more useful areas to look at cause and effect kind of things. Like a lot of people say you take probiotic, but will they really replace the gut microbiome? That is also another question which happens, because until one is able to really repopulate completely, the kind of claims people claim, drink this and you'll be okay. I think it's a very caution, because there are, I don't know how many microbes are there in our gut, and what we are giving is probably 10 to the power 5 or 10 to the power 6 cells. Will they
Yes.
be able to replace the rest of them is a fundamental question. That
Yes.
is why they say that, one of the suggestions they made is, when a child is born, if you don't do cesarean, the child might be better protected with the microbe than if you do cesarean and take them, because they may be prone to more. That's what gut microbiology also people are claiming that. So
Yes.
as far as possible, a normal delivery is important. The child will take all the microbes from the mother also.
We'll sir take this last question from Dr.
Yeah.
Neeraj himself. He is asking how this microbiome composition maintain in each generation. How it maintain generation to generation.
It is not true that they maintain generation, because there also mutations are happening all the time. Some of them are getting wiped out, new fellows are coming, just
Yeah.
like coronavirus. I mean, there are so many mutants appearing. RNA is only, if it is the RNA one, the mutation rate might be very high. DNA, it might be less. But otherwise, all of them are changing. Nothing is constant. I mean, Darwin was right there. There's a constant evolution happening to optimize.
Yes, I know about a study where gut microbiomes were tested in a community, and also within the family.
Yes,
Food habit actually determines the gut microbial population of the individual. But at the
correct.
community level, you may find a lot of common, species. But for
Yes,
example, one interesting study was the type of wine or alcohol the person consumes.
yes,
Right. So that's a very interesting study. You can find out, and even they tried to look, what is the microbial composition if a person consumes whiskey, and what is the
yes.
microbial composition when he consumes some other kind of drink like brandy or rum or some local,
No,
fermented wine. Because these alcohols are, sterilizing agents, and they wash off the gut microbe very easily.
Very easily, very true,
Yes.
very true. And whether they'll survive that much percentage of alcohol.
Yes. So, but now they have found out that these alcoholics are not totally having, their gut is not free. I mean, they also have a particular community. So,
Very true.
yeah.
Very true.
So I think this is a very emerging field still. We know very little of the this particular microbial genome. Okay. So let us stop here for today because
And only one point I want to
so
make.
please,
All of them
give-
should read about the work of Louis Pasteur, because
yes.
he really taught us how to analyze various diseases, various conditions, whether there's a disease in silk moth or, or cattle, whole lot of things he did on bacteria and viruses. I think it's a very good exercise now how to do experiment to confirm, because people ask questions, but I want somebody who sits and does a intelligent experiment and prove or disprove something. I think young faculty should read Louis Pasteur's model as a method of scientific method, scientific research as a
Yes.
case study.
Yes. So just a minute before we wind up, I can see Professor Celeste Singh logging in, who has a lecture in the evening. So just for the purpose of testing, he is there, and maybe I can introduce him briefly to you.
Yeah, sure.
Professor Celeste Singh, can you hear me, sir? Celeste, sir, can you hear me? He is logged in. I can see him in the panelist. Maybe he is doing some testing from his end.
Testing from his end, yes.
Yeah. Right. So let me put a message to him. Okay. Yeah.
Anyway, we managed to hold the time till
Yes, sir.
1:45.
Yes, sir.
Thank
Yes,
you so much, Utpal.
Yeah. Okay. So let me give a formal vote of thanks to Professor Krishnamurty Kannan for his extensive lecture on this topic on genome architecture and genome engineering. And I can see a lot of comments in the chat box. I mean, they like the talk. And thank you, sir, for the lecture. And you did it in a very short time. And I think it was really taxing on you, but in spite of your busy schedule, you give your consent. And I know you are very busy, and you are having one lecture also today in the evening.
Yes.
And you have to work extra hours. Right.
That's okay. It's
Yes.
a pleasure. It's my
Yes.
pleasure. It's my
So I
pleasure.
thank all the participants once again. And then Professor Kannan is always available over there whenever you feel you need, you have some other.
They can email me and ask me.
Contacting him. Yeah. So just Dr. Geeta is telling on I'm just reading this I mean, there are many good comments, but Dr. Geeta is telling, thank you, sir. Very productive time spent with you virtually. So instead of me giving you some kind of an assessment, I think participants' information is very
kind of them. Very kind.
I hope. Telling, sir, thank you for the informative session. Yes, sir. Thank you. Thank you. Finally,
Thank you.
let us now take a break for lunch, and we will be meeting at 2:45. I have my lecture, and I think it will be very tough time after Professor delivers us a good lecture. I think I have to go and cut down my lunch time and do a little bit of preparation. Thank you, sir.
Very modest.
Bye.
Bye.